Hello everyone. Today my topic is on quorum sensing in cariogenic biofilm. Basically, what is quorum sensing? How social interaction is very much important for humans to communicate with each other. Similarly, bacteria also communicate with each other by producing, detecting, or responding signals between them to produce this oral biofilm or various other virulence factors. So, coming to an introduction, quorum sensing is a communication mechanism between bacteria that allows specific process to be controlled, such as biofilm formation, virulence factor expression, production of secondary metabolites, and stress adaptation mechanisms such as bacterial competition systems, including secretion systems. And how this quorum sensing plays an important role in the oral biofilm. Normally, the oral biofilm is formed by the planktonic cells, which initially attaches or adheres to the tooth surface, and that forms a monolayer, and by cell division, it forms into a microcolony and then into a maturation and then into a dispersal. Here, the quorum sensing plays an important role in maturation and in dispersion. And coming to a themes in bacterial quorum sensing, there are three different themes. Basically, the Lux I and R in case of a gram-negative bacteria, oligopeptide two-component type in case of a gram-positive bacteria, which includes AIP, auto-inducing peptide, and TCSTS called as two-component signal transduction system. And in case of a gram-negative and positive bacteria, we have a Luxis encoded auto-inducer 2. Coming to the signals in bacterial quorum sensing, where the signals are very much important for the bacteria to communicate. So coming to the signals, we have AHL, which is an acyl homoserine lactone, in case of gram-negative bacteria acting as a signal. In case of a gram or positive bacteria, we have AIP and we have TCSTS, that is auto-inducing peptide and two-component signal transduction system acting as a signal. Coming to a gram-negative and positive bacteria, we have auto-inducer 2, which is basically called as a universal language. That's because it has an interspecies communication between them. Coming to a gram-negative bacteria and how does it work? So in this, we saw the Lux and I and R acting as a theme in gram-negative bacteria. Where the Lux I, it is an auto-inducer synthase which produces this acyl homoserine lactone signal that usually crosses the cell membrane. Where Lux R, it is a transcription regulator that binds with an AHL to activate the target genes. This usually takes place at high cell density but not at the low cell density. Coming to a gram-positive bacteria, in this we saw oligopeptide two-component type acting as a theme. So in this, it has two signals like auto-inducing peptide and two-component signal transduction system. And basically how it works comes on the future slide. Coming to a gram-negative and gram-positive bacteria, we saw an auto-inducer 2, which is also called as a universal language, such because it has an interspecies communication between them, and Lux is encoded synthase acting as a theme in gram-negative and gram-positive bacteria. And what are the key players in quorum sensing? So basically, we have three, where that is auto-inducers synthesis, and quorum sensing regulators. In case of auto-inducers, we have acyl homoserine lactone in gram-negative, auto-inducer 2 in gram-negative and positive, cyclic dipeptides, bradioxetin, and other type of auto-inducers. In auto-inducer synthesis, we have AHL synthesis, AI2 synthesis, and synthesis for other type of auto-inducers. In quorum sensing regulators, we have Lux R type regulator or Lux P or Q type regulators. And what is a negative regulation of this quorum sensing? Basically, an anti-activator protein or AHL degrading enzymes, furanones, which is structured mimics, and various bacterial components is used to manipulate this quorum sensing. Basically, especially quorum quenchers, transgenic plants, human hormone interferes with basic quorum sensing and other quorum sensing inhibitor compounds. So how do you basically interfere with quorum sensing? This could be interfered or this could be inhibited, or the communication between the bacteria could be controlled by inhibition of the signal generated or interfering with signal dissemination or blocking of signal receptors or inhibition of the signaling response systems. So by these four ways, we naturally basically interfere with quorum sensing. And yeah, in this slide, we could see MS Dhoni, James, Messi, and our own superstar Rajnikant. So all these characters are more unique in their own field and have more unique characteristics. Like same way, the Streptococcus mutans are more unique among various other oral bacteria. That's because it has a two systems, that is mainly two quorum sensing systems. One is COM-CD type, which is basically seen in gram-positive bacteria. Another one is a COM-RS type. So how this COM-CD occurs or works? Basically, when we saw an AHL in case of 1 gram negative bacteria, it crosses the cell membrane. But in case of a gram-positive bacteria, the AIP does not cross the cell membrane. It needs an ABC transporter for producing this auto-inducing peptide. And COMC signal precursor of it is a COMC signal peptide. When it reaches a critical concentration, it binds with COMD by autophosphorylation. This COMD that binds with the COME by means of phosphorylation in expense of an ATP. All this cycle usually basically prevent this genetical competence, basically prevent the bacteriocin production and the self-immunity. This genetical competence inhibition is a special or unique which is possessed by this Streptococcus mutans. But that is not done by COM-CD, it is done by COM-RS system. So COM-RS system basically will usually inhibit the genetical competence. So this Streptococcus possesses both COM-CD type as well as this COM-RS type. So it inhibits both this bacteriocin production, self-immunity, and the genetical competence. And why is very important, like we have to know about quorum sensing. That's because it is emerging as an integral component of bacterial global gene regulatory network and is responsible for bacterial adaptations in biofilms. And it has quorum sensing inhibitors. It is mainly used as a preventive and therapeutic tools for combating oral polymicrobial diseases and is used to study bacterial multicellular behaviors. So by understanding this quorum sensing and quorum sensing inhibitors, we basically inhibit various oral polymicrobial diseases caused by this biofilms and also open up various prospectors in treatment of various oral polymicrobial diseases. And these are my references. Thank you.